If both PCR and real-time PCR amplify DNA, why was real-time PCR invented? The difference is not what they amplify, it's when and how they detect the amplified DNA. Let's start with the conventional PCR. Like every PCR reaction, it amplifies a specific DNA sequence through repeated cycles of three main steps: denaturation, annealing, and extension. Ideally, during the exponential phase of PCR, the amount of target DNA approximately doubles with each cycle, producing millions of copies of the desired sequence. But here is the limitation. Conventional PCR does not normally monitor amplification while it is happening. The reaction must finish first. Then the amplified DNA is separated on an agar gel using gel electrophoresis. If a DNA band appears at the expected size, the target sequence has been successfully amplified. This means that conventional PCR mainly answers a qualitative question: was the target detected or not? The way the conventional PCR result analysis depends on the DNA bands compared to a DNA ladder, also called molecular weight marker. The ladder helps us to verify whether the amplified fragment has the expected size. Band intensity may provide a rough estimate of the DNA quantity, but it is not a reliable method for accurate quantification. Now, real-time PCR works differently. Instead of waiting until the reaction is over, the instrument continuously monitors DNA amplification during every PCR cycle. It does this by detecting fluorescence that increases as more PCR product is generated. Now, you may often hear two names: real-time PCR and quantitative PCR. In most contexts, they refer to the same technique, but real-time PCR describes how amplification is monitored, while quantitative describes what is monitoring allows us to estimate, which is the initial amount of target DNA. The fluorescence signal can come from different sources. For example, a DNA binding dye such as cybergreen, or a sequence-specific probe such as Tachman probe. As the fluorescence increases after each cycle, the instrument generates an amplification curve instead of a simple DNA band. One of the most important measurements in the real-time PCR is the CT value. This is the cycle at which the fluorescence signal crosses a predefined threshold. The earlier the signal crosses the threshold, the lower the CT value, and the lower CT value generally indicates that more DNA target was present at the beginning of the reaction, and a higher CT usually indicates less starting DNA. To analyze the results, unlike conventional PCR, real-time PCR does not require gel electrophoresis for routine detection. Instead, specialized software analyzes the fluorescent data, uses the CT value, and estimates the initial amount of the target DNA. So, conventional PCR is commonly used for cloning, genotyping, mutation detection, and confirming the presence of a DNA sequence, while real-time PCR is widely used for DNA quantification, gene expression analysis using RT-qPCR, pathogen detection, viral load measurement, and molecular diagnostics. So remember, conventional PCR amplifies DNA and usually shows the results after the reaction is complete. Real-time PCR or qPCR monitors amplification during every cycle and helps estimate the amount of starting target. Finally, RT-qPCR needs an extra step, which is converting RNA into cDNA before starting the reaction. Think of it this way. Conventional PCR takes a snapshot after the reaction is finished, while real-time PCR records the entire amplification process cycle by cycle, and that's why it can estimate how much target DNA was present before the amplification even began.